Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors from the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is our more open format portion of the podcast and can include topical interviews or discussions. My name is Darlene Sandoval, and I'm joined by my podcast co-host, Dr. Kirk Habiger and Dr. Kevin Williams. How are you all doing today?
Hey, Dar, how's it going?
Getting
Hello,
ready
hello.
for a holiday
Yeah.
week.
Yeah.
So
Absolutely.
we're recording this intro just a week or so after the ADA scientific sessions, and we had a really fun ADA live podcast recording Monday of the Sci sessions with Diabetes Care on air hosts Alice Chang and Mike Rickles, which was moderated by the Diabetes Core Update host Neil Skolnick. We don't know when this will be aired, but you all should keep an eye out for it. That was a pretty fun interview.
Yeah, it was a good time.
Yeah.
Yeah, it was a lot of fun.
So I was curious, who was the oldest friend or colleague or person that you've known the longest that you bumped into at ADA scientific sessions?
Yeah, you better differentiate because those are two different
That's
people,
true.
right?
That's true.
Yeah. Age is just a number.
Yeah. Kevin, what about you?
It was really interesting. So, when you're going through grad school early on, and also, I guess, going through your career, there's these papers you come across and become scientific heroes or celebrities in your mind. And I remember when I was in graduate school, my background is electrophysiology, studied leptin biology, and Michael Cowley published a paper in 2001 on leptin effects activating hypothalamic neurons. And I just thought that was an amazing study as a graduate student. I used it in the development of a lot of my hypotheses and my projects. And 2 years after he published that paper, I presented a poster at a scientific conference, and he came by my poster, and I was just so elated to meet the guy. And now, more than 20 years later, ran into him at the ADA scientific conference, and we were able to have some dinner and some drinks, and
Oh, nice.
just how your career progresses and the people that you interact with and communicate with is really cool. So
Yeah.
I guess I would mention Michael Cowley.
Cool. What about you, Kirk?
Yeah, I didn't see anybody from grad school, which means that my oldest colleagues are actually Darlene and Randy, right? So Darlene had to put up with me a couple times, and I ran into Randy Seeley watching a basketball game that did not go Indiana's way. So,
I'm sure he was giving you a hard time about that as well.
among many other things, yes.
That's right. That is his MO. I was just going to say that I didn't see anybody from graduate school either, but I did hang out with somebody who did a postdoc in the same lab as I did, Stephanie Gustafson. We got to have dinner. She works for a company and invited me to their company dinner. And so I got to be a thought leader, but also get to hang out with her and catch up. So that was pretty fun.
Very cool.
That's nice.
What was your question, Kevin?
Yeah, I have a question for you too. You mentioned it, Darlene, that we're coming up on holiday, 4th of July. We're just a few days out. And, there's a question that comes up around this time, and it keeps me up at night. And so I was wondering if you guys could kind of settle this debate on this question. Are you ready?
Ready.
We'll do it best.
I'm a little nervous.
Is a hot dog a sandwich or not?
Oh, wow. Huh.
What do you think? I mean, it's got bread, it's got a protein, it's got multiple condiments. You can make it whatever you want. Is it a hot dog or is it a sandwich?
I'm gonna go with yes, because
You
like
think
a
yes?
meatball sub is kind of the same thing, and it's a sandwich.
Yeah, I mean, subs or hoagies or
Yeah.
what have you are considered to be sandwiches.
What do
Yeah,
you
I think
think,
it's a sandwich. It's as much of a sandwich as a lobster roll or any other of these weird buns.
Bread plus meat plus
Yes.
condiment.
Yeah, absolutely.
Our
Yeah.
production manager is saying no. Thumbs down from Wyatt.
Yeah, I'm
You know,
surprised
3
it
out of
was so
4.
positive. We didn't split the debate, but maybe we do need to still put out a poll and have the listeners
Oh, yeah. Yeah.
give their opinions, see
Yes,
where it
please
falls.
put in the comments
Yeah.
for this episode.
How convinced were you?
Yeah. Is a hot dog a sandwich or something else? All right, Kirk, tell us what's going on in today's episode.
Absolutely. So to start, we interviewed July's paper of the month titled Novel Cell-to-Cell Communications Between Macrophages and Fibroblasts Regulate Obesity-Induced Adipose Tissue Fibrosis. This work was from co-corresponding authors, Dr. Takayoshi Suganami and Dr. Miyako Tanaka from Nagoya University. Then we chat with Dr. Richard Watanabe from USC to discuss his paper titled Genetic Variation in GCKR and PNPLA3 Regulate Metabolic Balance Across the Liver.
All right. And then Kevin, we interview, I think probably one of my role models in the diabetes field for Sweet Talk. Why don't you tell us about it?
That's right. This month we had the pleasure to chat with this year's recipient of the American Diabetes Association's Albert Renold Distinguished Career Award, who's Dr. Barbara Kahn. The award recognizes her outstanding contributions to diabetes research and mentorship. She really has led groundbreaking studies connecting basic molecular discoveries to real-world clinical impact and has mentored really a generation of scientists who continue to shape the field today. We're excited to talk to her about her scientific journey, her major discoveries, and her vision for the future of metabolic research.
All right. Another great episode. Let's dive in.
Next, we're going to discuss July's paper of the month titled Novel Cell-to-Cell Communications Between Macrophages and Fibroblasts Regulate Obesity-Induced Adipose Tissue Fibrosis. We're joined today by Dr. Takayoshi Suganami, professor in the Department of Molecular Medicine and Metabolism, the Research Institute of Environmental Medicine at Nagoya University. We're also joined by co-corresponding author, Dr. Miyako Tanaka, who is a lecturer in the same department. Doctors Suganami and Tanaka, congratulations on your paper, and thank you for being a part of this month's podcast.
Thank you very much for invitation.
Tradition with our podcast is to start with an icebreaker. And so the icebreaker question that I have for you all is name a favorite place you've lived and one reason why you like living there. So Dr. Suganami, we'll start with you.
Okay, I love Kyoto, Japan. I'm graduate from Kyoto University. I lived in Kyoto for more than 10 years.
Nice.
There are a lot of shrines, temples, and castles in Kyoto, and there are various historical places. So there is a very nice river named Kamogawa in the downtown of Kyoto. There are a lot of cherry blossoms along the riverside. We can enjoy jogging around there. So I love Kyoto.
That sounds lovely. Dr. Tanaka, how about you?
Thank you for inviting me to this interview. And I love Kyoto too, because my name is Miyako. Miyako is same meaning of Kyoto. My man loves Kyoto, so my name is Miyako. And Kyoto is very beautiful place to visit. And I was living in Kyoto for 4 years in my university student. It was so happy time. I enjoyed Kyoto. I enjoyed Kyoto very much. A very quiet place and a lot of temples and shrines. If you can get a chance, I recommend it to visit Kyoto in
It
Japan.
sounds lovely. I would love to.
I was going to say, I think it just moved up my list.
Yeah.
All right, Dr. Suganami, before we jump into the article, we'd like to give you a chance to highlight any other contributing authors that really helped this paper along.
Okay, I'd like to highlight Dr. Hiro Koda, the first author who introduced single-cell analysis to our lab as part of her PhD thesis work. This interdisciplinary study required diverse experimental expertise from single-cell analysis and electron microscopic analysis to clinical data analysis. So each collaborators brought essential skills for unraveling the complex cellular interactions underlying obesity-induced adipose tissue fibrosis.
Great. So now maybe you can give us an overview of your research program and then how this specific project fits in that one.
Our group is interested in understanding how obesity induces chronic inflammation in adipose tissue and other metabolic organs, leading to obesity-related metabolic diseases. Because adipose tissue contains various cell types with dynamic cellular interactions during the development of obesity, we focused on cellular crosstalk between parenchymal adipocytes and stromal cells, such as macrophages and fibroblasts. Our group was among the first to demonstrate that the vicious cycle between adipocytes and macrophages accelerates adipose tissue inflammation in obesity. This current project represents a natural evolution of our work. We introduced new technologies such as single-cell RNA sequencing and spatial transcriptomics to dissect the complex cellular interactions in obese adipose tissue.
Very nice. It really is a nice and large body of work that focuses on these crown-like structures in adipose tissue and the cellular mechanisms that drive the changes in those regions. Can you step back a little bit and tell us a little bit why adipose tissue fibrosis is a significant problem in obesity and why understanding those complex cell-to-cell communication processes is crucial for addressing fibrosis?
Sure. In general, chronic inflammation eventually leads to tissue fibrosis in various organs and diseases, and it's also the case with obese adipose tissue. Accumulating evidence has suggested that adipose tissue fibrosis is positively correlated with ectopic lipid accumulation, such as hepatic steatosis. Our previous work revealed that crown-like structures act as a driving force of obesity-induced adipose tissue fibrosis. They are unique histological structures in which macrophages surround dead adipocytes to engulf them. Activated fibroblasts also accumulate around crown-like structures. So there must be complex interactions between the cells in these microenvironments. Understanding this crosstalk is crucial because it could reveal new pathologic mechanisms for obesity.
That was a great background. Now I want to actually shift to Dr. Tanaka. In this work, you've utilized both single-cell RNA sequencing and spatial transcriptomics. For our audience, could you explain how these advanced techniques provide unique insights into this complex cell interaction that happens within adipose tissue and how traditional bulk-seq analysis might miss those interactions?
Thank you for your question. Crown-like structures are scattered throughout adipose tissue in obesity, and the characteristic macrophage and fibroblast subpopulations specifically located in crown-like structures are quite small. Traditional bulk RNA sequencing gives us average gene expression across all cells in a tissue sample, which may mask cellular heterogeneity. In contrast, single-cell RNA sequencing can analyze such small subpopulation as well as complex cellular interactions. Moreover, spatial transcriptomics can show us gene expression profiles in crown-like structures, the unique microenvironment accelerating adipose tissue remodeling. Using these new technologies together, we successfully identified novel macrophage fibroblast crosstalk that we never could have seen before.
Very nice. Dr. Tanaka, I'll stay with you. You identified a small subcluster expressing macrophage-inducible C-type lectin. Is that pronounced Minkle?
Yes, Miko.
Yes. Okay, good. That was specifically localizing to the crown-like structures. So can you elaborate on what Minkle is and what makes these Minkle-expressing macrophages particularly unique or interesting compared to other macrophage subtypes?
Miko is an innate immune receptor originally discovered as a pathogen sensor for Mycobacterium tuberculosis. And we have previously demonstrated that MINCO is selectively expressed in macrophages within clan-like structures and acts as a cell death sensor promoting adipose tissue fibrosis. And now, lipid-associated macrophages also play important role in obesity-induced adipose tissue inflammation. So, what is the relationship between lipid-associated macrophages and MINC-expressing macrophages? Our single-cell data clearly show that there are distinct subtypes in terms of morphology and gene expression profiles. Although lipid-associated macrophages have been intensively investigated, our work highlighted the novel macrophage subcluster expressing MINCO that was previously overlooked.
Very nice. So, what does this suggest about the functional roles and interactions of these MINCL expressing macrophages within the complex adipose tissue environment?
The key difference is location and abundance. Lipid-associated macrophages constitute a relatively large subcluster and are widely distributed in adipose tissue, including clan-like structures. In contrast, MINC-expressing macrophages are just a small subcluster, only 2% of adipose tissue stromal cells, and are selectively located in clan-like structures. So we think that lipid-associated macrophages play pro-inflammatory roles throughout adipose tissue, and MINC-expressing macrophages regulate tissue remodeling right around clan-like structures like local controllers.
Great. So just one more question, Dr. Tanaka.
Yeah.
In these findings, you characterize an axis of MINCL and MINCL oncostatin M, or OSM, where OSM secreted by the MINCL expressing macrophages acts on the fibroblasts to regulate collagen gene expression. Can you walk us through the significance of this specific signaling pathway and what it implies for the precise control of fibrosis in adipose tissue?
Tissue fibrosis is actually a dynamic and complex process in which both degradation and synthesis of extracellular matrix occur simultaneously. This active tissue remodeling is assumed to occur around clan-like structures. In this study, we found that oncostatin M, secreted by MINC-expressing macrophages, acted on the oncostatin M receptor on fibroblasts to suppress collagen gene expression by counteracting TGF-beta signaling. What is really interesting is that oncostatin M and its receptor were mainly expressed around clan-like structures. So we think that the MINCO oncostatin M axis plays a role in fine-tuning tissue remodeling around clan-like structures during the development of obesity.
So I guess just as a curiosity, if this fibrosis is developing during obesity, does weight loss or maybe some of our favorite anti-obesity drugs regulate the reduction in fibrosis via this same signaling axis?
Okay, that's an excellent question. Unfortunately, this study does not directly address weight loss scenarios, but we are currently investigating how adipose tissue fibrosis reverses during weight loss. Although it's difficult to reverse advanced fibrosis, our preliminary data shows that weight loss at the optimal timing for the appropriate duration can ameliorate adipose tissue fibrosis, thereby leading to better systemic glucose metabolism. Interestingly, we observed a transient increase of MINC expression during weight loss, suggesting the role of the MINC oncostatin M axis in reverse remodeling.
Interesting. Dr. Suganami, I'll stick with you. In the manuscript, you mentioned that crown-like structures are observed in other organs, such as the liver with metabolic dysfunction-associated steatohepatitis. I still haven't quite got that new language rolling off my tongue. And in the kidney with kidney disease. So, especially with obesity-associated kidney disease. So, could you discuss the potential broader implications of your findings for other fibrotic pathologies?
Okay, thank you very much for raising this important issue. Cloud-like structures represent sites of cellular interaction between dead cells and surrounding macrophages that engulf the dead cells, thereby causing chronic inflammation and fibrosis. We have found similar structures in the liver of MASH, metabolic dysfunction associated with steatohepatitis, and in the kidney during the process of acute kidney injury becoming chronic. Their histological features look similar between the organs and diseases, and they all induce chronic inflammation and fibrosis. In contrast, they have organ and/or disease-specific aspects. For example, MINC plays an important role in adipose tissue and kidney, but appears to function differently in the liver. We are excited to apply our analytical approaches to MASH and acute kidney injury.
Interesting.
Dr. Suganami, we started out with a discussion of where this project came from. So maybe we should finish with where it's going, right? Any hints as to where you'd like to take this work next?
Okay. This work really demonstrates the power of combining cutting-edge technologies with fundamental biological questions. We successfully identified a small macrophage subcluster expressing MINCO and revealed the complex interactions between macrophages and fibroblasts around cloud-like structures. However, our intervention experiments, such as bone marrow transplantation experiment, were not subcluster specific yet. For the next step, we need to use new intervention technologies, such as a combination of Cre-LoxP and Dre-Lox systems. In addition, as I mentioned, we are really excited to apply our analytical approaches to MASH and acute kidney injury because cloud-like structures are an excellent target for spatial transcriptomic analysis.
Awesome. Dr. Suganami and Tanaka, we want to thank you for joining us today and sharing your expertise and discussing your recent work on cellular crosstalk in adipose tissue fibrosis. I encourage the listeners to read the full article in the July issue of Diabetes.
Thank you very much for having us.
Thank you very much.
Next, we're going to discuss a paper titled Genetic Variation in GCKR and PNPLA3 Regulate Metabolic Balance Across the Liver. Today, we are joined by Dr. Richard Watanabe, Professor of Population and Public Health Sciences in the Department of Population and Public Health Sciences, Keck School of Medicine at USC. Dr. Watanabe, congratulations on your paper, and thank you for being a part of this month's podcast.
Thank you very much for the invitation. Happy to be here.
So, Richard, we're going to start off by throwing you off a little bit and giving you a question we didn't give you ahead of time.
Okay.
And we like to ask icebreaker questions. And so our icebreaker question for you is, who is the most inspirational scientist you can think of, live or dead, and why?
Oh, wow. I think I have two. One is Claude Bernard.
Nice.
Because when I first started doing science or thought of science as a career, one of the things my mentor sort of stressed was to go back and look at the old literature. And since I was interested in doing diabetes related research, I decided to go through kind of the history of diabetes. And of course, Claude Bernard is a major player in that history. But it just showed me how even back then, in the absence of what they knew, to sort of think through a problem and try to think of what experiments can be done to answer those questions that they had. I think the other thing that I found impressive was how much that ancient work keeps getting cited and keeps getting rediscovered. As we start applying, more modern technologies and are able to do things that they were never able to do.
Yeah.
And then I think my other inspiration is Francis Collins. I've had the great pleasure of working with Francis for, I don't know, maybe 30 years or so in the field of genetics. And I think one of the things I've learned from him or continue to try to learn from him are better communication skills. Because this is somebody who can take probably the most complex scientific things and explain them to anybody in a comprehensible way. And that's a very unique skill.
Yeah.
And I find it always challenging. My parents still don't know what I do, no matter how many times I've tried to explain it to them. I probably should have them talk to Francis, but anyways, I find that skill unique and something that I aspire to obtain and utilize, because I think even more so today than ever, I think scientists have a real responsibility to be out there explaining the complex things that we do to the public,
Yeah.
so
Sure.
that they can understand it and understand why the work is important and why, they should be doing things or not doing things.
Yeah. Couldn't agree more.
That was a great answer.
Hmm.
So, Richard, I want to give you a quick platform to name anybody, collaborators, students who may have contributed to this work that we're about to talk about.
Well, like any other scientific team, I mean, everyone made contributions, and this work wouldn't exist without everyone's help. But I wanted to highlight the students on this project because there are quite a few of them. But I think unlike a lot of other scientific projects out there, the vast majority of them are undergraduates. So the two co-first authors, Young Ma and Shi Zhou, were master's students in our department, getting their master's in biostatistics. And they sort of split the work and, kind of did half and half. However, the undergraduate students, so Felinda Lowe, although the name is spelled TH, it's actually pronounced with an F.
Ah.
So it's Felinda Lowe. And David Fan and Tyler Finley did a lot of the critical early work on this project to help convince me that what I was thinking was right. I had some questions about, whether how I was interpreting things was really how things were working. And they put a lot of time and effort into not just doing literature searches, but also actually doing data analysis to help bring everything together. So those 5 students deserve a lot of credit.
Yeah, that's always great to be able to bring in the young voices, right? Now let's start by giving an overview of your research program, right? And kind of how this project specifically grew out of it.
So my research program basically can be broken into 2 broad categories. One is genetics, and primarily is kind of the gene discovery work, right? Trying to identify loci that contribute to risk for diabetes and obesity, and some of the more recent molecular work in trying to find the functional variants out of the things that we've identified either through genome-wide association or sequencing studies. That probably formed, I don't know, maybe the last 30 years of my research career. But before I got into genetics, I'm formally trained as a systems physiologist. So I used to do systems physiology before that. And when I came back to USC after doing my postdoc at the University of Michigan, I decided I would go back to those roots and start doing clinical physiology with the goal of trying to understand how these genetic variants we've identified alter physiology to confer risk for these diseases. And so we do basically mechanistic clinical studies based on these genetic variants and trying to understand the mechanisms they alter and how they might change things.
Nice. So let's jump into this specific project. So here you were focused on 3 risk variants for type 2 diabetes and type 2 diabetes related traits. And those are their risk alleles associated with glucokinase regulatory protein, and palatin like phospholipase domain containing 3. So, can you provide a little bit of background on these variants, what risks they're associated with, and why you were interested in the physiological consequences of these particular variants?
Right. So it's kind of interesting. So glucokinase variant, not just one variant, not the one that just the one we studied, but there have been a number that have been shown to be associated with diabetes risk and diabetes related subphenotypes. And that makes sense from a, straight up candidate gene biology perspective, right? Given the role glucokinase plays. Glucokinase regulatory protein is interesting because it actually regulates glucokinase in the liver. So those two proteins, and actually glucokinase regulatory protein is one of the weird gene naming conventions because actually it encodes for a protein that's called GKRP, glucokinase regulatory protein. The gene name is glucokinase regulator. It's like one of a minority of gene names that don't translate directly to the protein names as being the same. So, but basically, what GKRP does is it sequesters glucokinase, binds to it and sequesters it until glucokinase is needed, and then dissociates and allows glucokinase to go do its thing. So it made sense to be wanting to look at those two because of genetic variation in GCKR also is shown to be associated with the risk for type 2 diabetes related phenotypes, but also with steatotic related phenotypes, liver fat itself, liver enzymes, etc. And then PNPLA3 is interesting because it's only been shown to be associated with liver fat based phenotypes, liver enzymes, steatosis itself. It's never, at least to my knowledge, it's never been shown to be related to any other disease or disease related phenotype. You know, the literature up until this point basically seemed to anchor it to steatosis. So we wanted to look at these because all of three of these are present in the liver. They all play critical roles that seem to be tied to steatosis. And people always used to ask me, well, why GCK and GCKR? But if you think about it, you know, those are regulating glucose entering the liver. And then depending on metabolic state, right, the fate of that glucose can go in a bunch of different directions, fat being one of them. It was this idea about, well, you've got these things that are kind of regulating, you know, substrate uptake by the liver. You've got another one that's sort of regulating the breakdown of fat. That's PNPLA is a lipase. So it's basically taking the fat and converting it to triglyceride for release. And so it's kind of like regulating the balance across the liver, if you will. And that's sort of where the idea came from and why we focused on those three.
So I wanted to dig a little bit deeper while we're talking about biochemistry here. Can you explain the connection between fructose and glucokinase and the glucokinase regulatory protein?
Yeah. So as you know, we don't endogenously produce fructose, but we can metabolize it, and it's primarily metabolized in the liver. It's been known through in vitro experiments and other non-human experiments that fructose metabolites basically regulate glucokinase regulatory protein. But that, to our knowledge, had never been shown in vivo in humans. And so that was our interest in looking at that because, as you know, there's all this evidence showing that high fructose is metabolically bad. It's known to contribute to steatosis. It's known to contribute to type 2 diabetes. There's way too much fructose in our food supply. And my thought was, well, this could help explain why fructose might be metabolically bad. I mean, it's like anything else in the world, right? I mean, a little fructose is good. Right. Too much fructose, no bueno. And so, trying to understand that mechanism. And the fact that nobody has, shown, and we all speculate that that's what's happening, that what happens in vitro was happening in vivo. But I think this is the first time we've provided evidence that it might be true in vivo.
Yeah. The dose makes the poison, right?
Yeah.
All right. So now taking a complete 180, right? For going from biochemistry to the population you chose to study, how did you hone in on the Mexican American, Hispanic American population and the risk variants for type 2 diabetes?
This comes from a long tradition here at USC. So Tom Buchanan, Annie Jiang, and I have been working in this population I don't know, ever since I was a student, actually. Tom Buchanan basically was the one who sort of started our kind of diabetes group here that's been looking at this population. So his interest has always been in gestational diabetes, and the role of gestational diabetes in the development of type 2 diabetes. And, he was the first one to show, at least in this population, that women with previous gestational diabetes were at elevated risk for developing type 2 diabetes. They had a postpartum 5-year risk of 55%. And he's been interested in trying to understand what that mechanism is that elevates their risk. And at one point, he and I talked and said, well, we should, we've been focusing on the physiology and et cetera. We should probably try to look at the genetics. And this was the reason for this, what we call the beta gene study, which was to go in. We went in and collected Mexican-American families of probans with and without a previous diagnosis of gestational diabetes. The idea being that by grabbing them, we should be able to capture the spectrum of at-risk individuals without actually picking up people with, frank type 2 diabetes, and be able to study an at-risk population by, providing detailed phenotyping and then looking at genetic variation.
Yeah, so that was going to be my next question is to kind of talk about and describe this study recruitment and the specific types of phenotyping experiments that you've performed.
Yeah, so we're kind of different from a lot of other groups. We do fairly detailed phenotyping, mostly because we understand that a lot of these things are very interactive. Quite frankly, trying to get a good handle on different aspects of the metabolic profile was important. So we do DEXA scanning to get body composition, primarily to get body fat. And one reason we do that is because it's been our experience that BMI is not sensitive enough to be capturing interactions with adiposity. I'd love to be able to do like whole body CTs on everybody, but that's not affordable. So we do DEXA scans. And then we do oral glucose tolerance tests with 30-minute sampling. So we're basically taking samples every 30 minutes over the 2 hours. Sometimes we extend the experiment out to 3 hours. And the idea there is to get a good handle on oral glucose stimulated phenotypes. Again, knowing the fact that the incretin effect plays a role, and that's something we wouldn't capture if we were looking just at intravenous glucose. But then looking just at oral glucose has problems because the oral glucose tolerance test in and of itself has some issues in terms of absorption rate, whether the participants actually, drink the entire solution, which is sometimes a problem. So then on top of that, we do the intravenous glucose tolerance test with Richard Bergman's minimal model to get measures of insulin sensitivity, insulin secretion, glucose effectiveness, which was one of the primary outcomes for this particular paper, and then looking at disposition index as a measure of beta cell function. So we try to capture an entire metabolic profile on these individuals, in addition to more classic fasting blood-based things like lipids, et cetera.
Great. So now let's dig in a little bit to the results here. We know that these genes are all important for liver metabolism, but there are different degrees and types of associations between these specific variants and specifically type 2 related phenotypes. Can you tell us a little bit about your findings and specifically those interactions?
Yeah, no, just looking at the individual variants and their associations with the individual phenotypes really didn't, teach us anything new. We pretty much replicate what others have already seen on a genome-wide basis, from genome-wide studies. I think the unique outcome that we found just by the straight-up associations was the association between the variant in GCKR and glucose effectiveness, because that's a phenotype that very few people have. So we were able to introduce that as a new outcome, but that was also then the genesis of looking at the interactions either between the variants, but also the interactions with dietary fructose levels. And so we were actually able to show that that particular GCKR variant does interact with dietary fructose, such that the greater the fructose level, the less effective GCKR is and therefore the liver takes up more glucose. So you can actually, if you look at the figure in the paper, you can see the effect of fructose to increase glucose effectiveness. But then within each, well, we broke it down into tertiles for display purposes. But within each tertile, you could see the effect of the genetic variant to differentiate glucose, SG glucose effectiveness, even within those tertiles. And so there does seem to be this effect, which is very important, we think, because then it sort of tells you how much glucose the liver may or may not be taking up, and whether that energy is then being diverted towards, the development of steatosis.
Right. And I guess just as a quick follow-up, these variants, were any of them coding, or do they directly regulate expression, or?
So this is probably something I should have mentioned earlier when you asked about my research program. We're working with variants that are simply being reported out of the genome-wide associations or the sequencing studies.
Right.
These particular variants we're looking at are non-coding. I take that back. I'm sorry. The PNPLA3 one is.
Okay.
But there's no evidence that they're functional variants. Let's put it that way.
Okay.
And basically, my principle has been that if we believe these little signposts, right, these signals are supposed to be in linkage disequilibrium or somehow correlated with the functional variant. And if we believe these signals, then these variants should be sufficient enough for us to stratify individuals to try to understand the biology. And so I'm relying on somebody smarter than me to go and actually find the functional variant, but at least we'll have the physiologic data to help explain how these things may or may not be working. And then getting back to the results, the other unique thing that we've found is with PNPLA3, as I mentioned previously, that seemed to be kind of restricted. The associations that have been identified seem to be restricted only to steatosis related things. And we showed that if you look at the interaction between PNPLA3 and GCKR, we can start to see associations with other like diabetes related phenotypes. So we start to see associations with fasting insulin, 2-hour insulin. We have a marginal association with insulin sensitivity. So those three kind of tie together. And so it does tell you that looking at interactions amongst genetic variants or also environmental factors can start to teach us new things about these gene products and what they may or may not be doing.
All right. So what do you think the broader implications of these findings are? Do you think screening folks for these variants might be helpful in directing, maybe for physicians in directing care, or in patients thinking about how they should be more careful with fructose intake, et cetera?
Yeah, I think it's a little premature for that, as much as I would love it. But I do know, for example, GCKR based drugs are being tested. And same is true for PNPLA3 based drugs have been tested and are showing promise, at least in terms of steatosis. It does, I think, from like the 100,000 foot level, it does tell us at least that people, given how bad our food quality is, keeping track of how much fructose you're actually ingesting is something, at least from a dietary, lifestyle modification thing, that everybody could be doing.
Yeah.
We need to be doing more research on this. In fact, I just finished a grant where, because what we've been looking at has just been liver enzyme levels and not actual assessments of liver fat. I just completed a study where in a smaller group of individuals, we went and did MRI-based fat measurements. And we'll be testing the variants first, see if we could replicate what we found now with actual liver fat. Also, we're thinking about designing experiments to resurrect some of these old protocols we did many, many years ago using labeled glucose to actually measure actual glucose uptake, because glucose effectiveness is only reflecting liver glucose uptake. It includes a bunch of other things. And so to do some actual metabolic studies to actually measure glucose uptake by the liver and also try to assess the fate of those glucose molecules and see whether they're actually going to fat or whether they're going to glycogen or wherever they may be going, to really understand these mechanisms.
Yeah. Are there any other key points or thoughts you'd like to leave us as we close today?
For your younger audience, systems physiology is not dead. I think systems physiology
You're,
has
at
much.
least with me and Kirk, you're
That's
preaching
right.
to the
Yeah,
choir.
okay, that's good. No, because as I like to tell them, knowing the functional variants in these genes is important, but because glucose is one of the most highly regulated things in the body, probably the most regulated thing in the body, a lot of these, the metabolic effects of these things are not gonna be reflected in something that can be clinically measured. And we need to understand how that works or how it doesn't work. I mean, one of the things that Tom showed in his cohort of Mexican American women, and it's also been shown in the Pima Indians and a couple other groups, if you follow at-risk individuals longitudinally, their glycemia only creeps very modestly, and then something breaks and suddenly the glycemia goes up, right? And so we need to understand what's going on so that we can figure out what to measure while the glucose is just creeping.
Yeah.
Right? By the time the glucose starts changing dramatically, it's probably a little too late.
Yeah. Very nice.
Well, Richard, we want to thank you for joining us today and for sharing your expertise, and especially for discussing your recent work on these genetic variants, their role in hepatic and whole body metabolism. I encourage our listeners to read the full article, which will be in the July issue of Diabetes.
And thank you very much again for the invitation.
Welcome to another edition of Sweet Talk. I'm Kevin Williams. Today, we're honored to be joined by this year's recipient of the American Diabetes Association's Albert Renold Distinguished Career Award, who is Dr. Barbara Kahn. She is also a past recipient of the ADA's Outstanding Scientific Achievement Award and the Banting Medal, making her one of a select few to receive all of these prestigious honors from the American Diabetes Association. I'm also joined by our co-hosts, Dr. Darlene Sandoval and Kirk Habegger. Dr. Kahn is the George Richards Minot Professor of Medicine at Harvard Medical School and Vice Chair for Research Strategy at Beth Israel Deaconess Medical Center. Her pioneering research has uncovered key molecular mechanisms contributing to type 2 diabetes and revealed how adipose tissue regulates systemic insulin sensitivity. Dr. Kahn, welcome to Sweet Talk.
Thank you very much. It's a pleasure to be here.
We're definitely excited to celebrate your remarkable career and explore both historical milestones and future directions in the study and treatment of health and disease. But before we get into it, we usually like to start off with a little bit of a fun icebreaker question. And so the question we have for you is, if you were not focused on your current research, is there another career or field you could imagine pursuing?
I've thought about that question. And honestly, I don't think there is another career that would be equally rewarding. I think if I had it to do over again, I would get the MD. I'd do the clinical residency and fellowship, and I'd do the basic science training at NIH again, because scientific discovery grounded in clinical relevance really provides a very rewarding, gratifying career. However, I would say if I'm advising young people now who come to me and say, should I go to medical school or get a PhD or pursue something else, I really listen closely to their goals. Because now I think for some people, careers like bioengineering or biostatistics or other disciplines might be a more suitable choice than the long haul of the MD and basic science training.
Barbara, your work has shaped the way that we understand insulin resistance, and specifically through the lens of adipose biology. You've been at the forefront of some major shifts in the field, both conceptually and mechanistically. And before we get to the science, I'd love to start with your story. So what first drew you to the fields of metabolism and insulin resistance? Was there a defining moment or an experience that you can share with us?
Well, I could start with, why did I even decide to go to medical school? And then why did I decide to go into scientific research? So I can remember back to when I was a kid, and my grandmother would have these debilitating headaches, and everyone had to tiptoe around. And of course, as kids, we were always slamming the screen door, and it was so I
Yeah.
really felt, isn't there a way to know more about what's going on so we could find a treatment for these debilitating headaches? And then when I was a freshman at Stanford, the eating disorders were rampant in the women's dormitories. And again, I really had that strong instinct to want to understand the physiology and the pathophysiology. So I eventually went to medical school really to understand how the human body works and what goes wrong in disease. And at the end of my residency, I realized I couldn't just hang out at Shingle and go into practice, in an office somewhere. I really needed to be involved in finding new information for either diagnosing or treating disease. So, as a general medicine fellow, I did a project on the comparison of home blood and urine glucose monitoring. And this was county hospital, and we recruited the patients from our clinics, and every patient who entered the study got better, whether they were on home blood or urine glucose monitoring. And I realized it was a study effect, when you get the attention and the input and the interest from health professionals, you can improve your metabolic control. So I went to the UK to compare our results because they were reporting in small, uncontrolled studies that people on blood glucose monitoring did better. And when I went to these different centers in the UK, they took me on tours around their basic science units. And I realized diabetes did not have to be so depressing. And really, basic science was the way to make breakthroughs in treatment of diabetes. So I decided to do an endocrine fellowship. I applied widely. People strongly suggested I go to the NIH. I went to NIH and I interviewed with Jesse Roth, who, was the king of insulin receptors. And Jesse introduced me to many different scientists. And then he would hear my feedback. And in the end, I decided I was totally captivated by how insulin elicited the translocation of glucose transporters from intracellular pools to the plasma membrane. And that if we could figure out how to augment that or get over the block that exists in type 2 diabetes, we could make people more insulin sensitive. So that's my story of getting to insulin action as my career.
So, one of the aspects of your work that I think has been most transformative is it's really impacted how we think about adipose tissue, right? So, we used to sort of think of it as this passive storage depot, but your work has really framed it as more of a dynamic endocrine organ that plays a role in metabolic health. Is there any particular finding that you feel has had the I'll put it two ways. I'll give you choices for either end, whether it's a finding you had that had the biggest impact of the field or a finding you had that you think should have had a bigger impact that maybe hasn't quite been recognized yet.
Well, I think the most important finding was early on. So, as you mentioned, in the 80s and 90s, fat was considered an inert storage depot. And we were very intrigued. When I first started working on glucose transport, the glucose transporters had not been cloned. it was thought that there was one facilitated diffusion glucose transporter. There were other sodium-linked glucose transporters, but just one glucose transporter that we measured by cytoclasin B binding. So it was not known that there was anything really special about insulin sensitive tissues, except that when you did the cytoclasin B binding assay, there was more movement of those transporters to the plasma membrane in response to insulin. So when I started my faculty position, Mike Meckler had just cloned GLUT1 in Harvey Lodish's lab. And everyone thought that was the glucose transporter. But it didn't really, the data with GLUT1 by Western blotting didn't really parallel the cytoclasin B binding data. And so others in the field started looking for other glutes, and eventually GLUT2, 3, 4, etc., up to GLUT14 were cloned. So we did some of the early evaluation of the regulation of GLUT4 in obesity and diabetes. And we did this in rodent models and in human adipose tissue and muscle. And we saw in both the rodents and the humans that GLUT4 was downregulated in insulin resistance and type 2 diabetes. So that made us decide to knock out GLUT4 specifically from fat cells. And this work was done in the early 90s. And actually, Bruce Spiegelman had cloned the AP2 promoter, and he had used it with diphtheria toxin to ablate adipose tissue. But no one had actually overexpressed, a metabolically functional gene in fat. So Peter Shepard, my first fellow, overexpressed GLUT4 in fat, and we really did not expect it to have the profound effects that it did, which were a modest level of obesity, adipocyte hyperplasia, and marked improvement in glucose tolerance. And I think showing that adipose tissue and glucose transport in adipose tissue could regulate systemic insulin sensitivity was really an important finding.
Very nice.
And Barbara, you particularly have consistently bridged basic science and clinical relevance. And I was curious, is there a particular approach that you have into translating your lab discoveries or discoveries at the basic science level into something that impacts human health or therapeutics? Do you think about translating these basic discoveries in the lab into something that impacts human health or therapies?
Well, if we discover a new molecule that looks interesting in the lab, before we really dive into in-depth studies, I always validate it in humans. So if that doesn't exist or it's not regulated in humans, then we don't pursue it. The clinical relevance is really important for me. Yeah. Right now, I am very much trying to get these fatty acid, hydroxy fatty acids that we discovered. And incidentally, we discovered those from the adipose GLUT4 overexpressor model. We're very much trying to get those into the clinic. And originally, we thought they'd be really beneficial for type 2 diabetes because they augment glucose stimulated insulin secretion from human islets, and they promote glucose transport in adipocytes, and they reduce hepatic glucose production. But I didn't really want to compete with the incretins, so we're now focusing on type 1 diabetes because we've also shown in mouse models that these lipids profoundly prevent type 1 diabetes in autoimmune mice. So we're hoping to be able to do at least a proof of concept in people.
And in steering your direction in research, you mentioned in the opening the difference between the controlled and the uncontrolled studies and what was being found in humans in their blood glucose regulation. I was curious, are there any other clinical observations that might have surprised you? Maybe they helped inform more about what was going on in your models and your systems as it regulates adipose tissue and insulin sensitivity.
Early on, a lot of people in the insulin action field were studying the insulin receptor. And undoubtedly, we have learned a tremendous amount in studying the insulin receptor and its kinase activity and so on. But in people and in mice, you can reduce the number of insulin receptors over 90% in fat cells and still get normal insulin action. So I think it was a surprise that the glucose transporter was really more important, molecule for molecule, maybe than the insulin receptor. So, we collaborated with Kahn on the paper where he knocked out the insulin receptor from adipose tissue. There was an odd phenotype with changes in the adipose tissue, but not really profound insulin resistance. Whereas if we knock out GLUT4, the animals are quite insulin resistant. I think that also people didn't realize that lipodystrophy would be associated with such profound insulin resistance. But I think the most severe diabetic people are people with lipodystrophy.
That's really fascinating.
I could also say that my very first science paper was actually on the TSH assay, thyroid stimulated hormone. And that was completely informed by a patient that I saw at the NIH. And this woman had Sjogren's disease, an autoimmune disease, and she was referred to the endocrine clinic because they thought she was hypothyroid because her TSH was 10 times elevated or more. And I looked at her from across the room and I knew she was hyperthyroid, not hypothyroid. And all the other tests pointed to hyperthyroidism, as did the physical exam and the history. And in the end, what I figured out with one of the clinical chemists at NIH was that her autoantibodies were interfering with the TSH assay
Wow.
and falsely raising it. So as a result of that, all the TSH assays at the time were revised. And so patients can really inform, basic research.
Wow. Yeah.
That's really cool.
So Barbara, I'm hoping maybe you can look into your crystal ball now, right? And you mentioned the incretins, and you mentioned your work on, and if I get this wrong, I'm sorry, the fatty acid esters of hydroxy acids, right? Aside from those two as future therapeutics, is there something that really excites you? Is there a pathway that you think has promise, especially, I guess, in terms of insulin resistance and type 2 diabetes?
Yeah. I mean, the incretins are very impressive. And, the dual and tri-agonists are incredibly impressive. And, the fact that glucagon agonism in combination with other incretins is so beneficial is surprising and really interesting. I think the key there is that rapid weight loss is associated with lean body mass. I don't want to get into the argument of is it out of proportion to other kinds of weight loss. It's just a fact that when you lose a lot of fat, you also lose lean body mass. And if people have to be on these drugs continuously for decades, that could be a concern, especially as people lose lean body mass with aging. So I think, to combine some of these TGF beta family agonists or other molecules that would build muscle would make the incretins even more powerful as a long-term approach. There are other anti-obesity approaches that are under investigation, and some of them are better tolerated. And, I think back to the days of the PPAR gamma agonists and, troglitazone, sold as Regulin, was the most potent. And it was the first to be taken off the market because of very rare but true, liver toxicity. So I don't know that the absolute most potent agent is in the long term going to be the safest and the most durable agent. But I guess you were asking me more to come up with something really novel. And what I would say, where I see incredible promise right now, is for type 1 diabetes. And the idea of being able to prevent and even reverse very early stage type 1 diabetes. I think these immune modulators are really intriguing. And I think that, we again could get to something even safer that could be given weekly or monthly or in a more sustained fashion. It's terrific. They're preventing type 1 diabetes for 1, 2, up to 5 years. But maybe you could prevent it for decades with an even safer agent.
Yeah, well said. Well said. Do you think that there's any room for direct targeting of fatty tissue, of the adipose tissue? That's something that we've not really, I guess, specifically come up against since the TZDs, right?
Right. I do think there are promising approaches to increase insulin sensitivity and prevent or treat type 2 diabetes. For example, increasing secretion of beneficial peptides or lipids from adipose tissue, or decreasing the secretion of detrimental ones. I also think the data showed that if we could increase de novo lipogenesis in adipose tissue, that would increase insulin sensitivity. For obesity treatment, I think it is tricky.
To a certain extent, the brain is going to respond and increase appetite. If you try to remove fat with liposuction, it grows back. If you destroy fat, in lipodystrophy, you get insulin resistance. So I don't know. I mean, there are several different companies working on brown or beige fat, and that might have promise. Of course, if you end up increasing heart rate and blood pressure, it's not going to be a durable therapy.
Right.
So I'm open-minded about that idea.
But to increase insulin sensitivity and prevent or treat type 2 diabetes, I do think there are promising approaches targeting adipose tissue directly.
Fair enough.
So we've talked about your impact on the field, at least conceptually. What are the biggest changes you've seen to basic science research, or even just focusing on diabetes and metabolism from a technical perspective? So what are those technical advances that you think are making or have made the biggest impact on the field?
Yeah, so many. It's really amazing. I mean, I hate to date myself, but when I was starting my lab as an instructor, that's when PCR was discovered. Can you imagine? We didn't have that before. Um, I think that the molecular techniques are just so much more, powerful now than they were back then. I think that the human genetics is a really major advance. And now genetic editing is so exciting for diseases where a single gene is known and the mutation is known. I think that it also wasn't quite as popular to do really multidisciplinary research. And for me, it's been so exciting to collaborate with chemical biologists and structural people, and people, outside of even biology per se. So I think the pace of discovery and the pace of technologic advance is really amazing and will probably continue. Bioinformatics and being able to deal with big data didn't exist back then.
Yeah.
And we touched on mentorship a little bit earlier in the aspect of mentors for you. And you mentioned that there were a few, 3, 4, 5 mentors that you could probably list that were huge support for you. And I know that you've been a mentor for a lot of these next generation of scientists, and many trainees have gone through your lab, through your stewardship, and on into independent careers. And I was curious if you have any kind of particular philosophy when it comes to mentoring. What do you think makes for that lasting mentor-mentee relationship?
Yeah, I think the first thing is to really meet the trainee where they are. So, the same style might not work for everyone. I also find that if I can help a trainee figure out what they are really excited about, it's more likely that project will work out than if I just superimpose a project that I'm excited about, but they maybe don't think is so great. So, I mean, it's very important to give guidance and so on, but also to sort of figure out what excites the mentee. One thing that I say when people from women's groups ask me, don't think there's going to be one person out there who's going to be your dedicated mentor. You sort of need to develop a tapestry of people who are supporters and mentors. It's kind of changed, and people are really busy, and there's a lot of demands in, academic life. So the best thing is to have kind of a group of people that you call on individually for different aspects. I think as a mentor, if you can get certain funding for people to bridge them when they don't get their R01, and important support during those really vulnerable periods, that can really pay off, because once somebody gets an R01, they have so much more confidence and see themselves as being able to be independent.
So I think the, science funding and support ebbs and flows right across the decades, and I've certainly seen some ebbs and flows, and we're in a little bit of a tip now. Is there any particular advice you would give to young scientists that are, ready to jump in and start a lab, and on one hand very excited about the promise, but concerned about dipping funding and how they're gonna
Yeah.
keep things going?
My answer to that is probably different than it would have been 6 months ago because of all the current events. So, if your institution allows it, definitely look for foundation funding, because it may not be as uncertain as what the NIH policies are going to be. I do think that many NIH study sections really look favorably on multidisciplinary, multi-PI grants, and to consider that, because you can really make your project unique if you have a unique combination of other PIs that bring other expertise. There's a great desire to publish in the top journals, and it is rewarding. But I would say to people starting a career, if you don't get it immediately into the top journal, get it published somewhere else, because you need to show that you're publishing, and people will find that paper, and you can hold out for a top journal later in your career. I also would say if a paper gets rejected, Burson and Yalo won the Nobel Prize for radioimmunoassay. Their paper describing the insulin radioimmunoassay was rejected by the journal Clinical Investigation. And then even the first resubmission was not accepted. And yet a Nobel Prize was given for that work. So persevere and, try to, carefully read criticisms, take into account whether they're valid. If they are, address them. If they're not, go elsewhere. Don't get discouraged. It does require resilience, but this is a very rewarding career.
Do you have any distinct, I mean, I think that advice really applies to everybody. Do you have any other more specific advice for young women?
In some ways, it is much harder for young women. In some ways, we've come a long way. We can't really talk about DEI now, but I know search committees I've been on and so on, there's certain requirements now in terms of having a broad scope in the candidates who are considered. But in the year 2000, when I became chief of the endocrine division, I was the only woman clinical chief. And there were actually about 25 people in the monthly division chiefs, because we had a division of signal transduction and a few non-clinical divisions as well as clinical. And I was the only woman there. Fast forward to last December, there was still only one woman division chief. Now, there are 2 women vice chairs of the department, but it's really, the progress is difficult, even though our institution has now a committee dedicated to the advancement of women, and they've done a lot of negotiating for, salary, for childcare, for various benefits, for support in terms of leadership positions and promotions. There's a lot of progress that's taken place on a lot of levels, but it's still, for the real leadership, it still takes a conscious effort. One piece of advice, I think institutions where women have these either formal committees or informal groups where they read each other's grants, they read each other's letters, they give feedback. I think those have been particularly successful.
Thank you, Dr. Kahn, for joining us today. I know this has been a real treat for all of us to sit down and chat with you. And congratulations again on the Albert Renold Distinguished Career Award that you'll be receiving at the ADA meeting. We're recording this early in June, so it'll be later this month. To our listeners, thank you for tuning in to this episode of Sweet Talk. As always, please like, follow, and subscribe for more diabetes bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.